about my research group called SMILE for Stochastic Models for the Inference of Life Evolution. And so this is the group a few months ago. And so what we do in this group is to conceive innovative models, or we try to predict patterns. So that's the math part, and infer processes. That's the statistical part. And as you said, Tom, we're interested in various topics, from the smallest scale to the largest scale. We're interested in somatic evolution, and notably in tumors. We're interested in epidemiology and evolutionary epidemiology in particular. And so we've been quite busy, like maybe less than you were in the last few years. We're interested in demographic inference from genomic data and applications to conservation biology. We're interested in modeling speciation by individual-based models. In the inference of coevolution between loci, characters, or species. And finally, in macroevolution, so the origin of species, if we can say rapidly. And of course, by math problems that are inspired by all these topics. So today, I would like to talk about the inference of macroevolutionary processes, which are the processes that have generated biodiversity as we know it. So the kind of processes that we're interested in are among the following ones, and there are others. Adaptive radiation, like the colonization of a new niche and the appearance on a short paleontological timescale interval, a short brief appearance of many species, inferring the various geographic modes of speciation that depend on the distribution of the mother species, the geographical distribution of the mother species. Divergent adaptation, which means adaptation to two different habitats where selection pressures are different. Mass extinctions. Species selection, which is the ability of some species to give rise to more species by speciation than others. And introgression from distant lineage, which is now a very well documented phenomenon. And so the most popular approach in recent years is called the phylogenetic approach to diversification. And in this approach, you assume that the diversification of species has unfolded through time, like a birth-death model, where the particles are the species, deaths are extinctions, and births are speciations. And the goal is to infer how speciation and extinction rates may depend on absolute time, on one specific clade, on the co-occurring diversity, which is called density dependence in population dynamics, upon the age of the species, or a trait like, for example, body mass. And the method is likelihood based on a single phylogeny. So here's a cartoon that I think was taken from a talk by Tanya Stadler, where you see on the left time going downwards. You see the diversification process and the tree that it has generated. Of course, so the blue. Yeah. Okay, so the blue segments are speciation events, and the red crosses are extinction events. The yellow round circles are sampling events. And so, of course, you don't know the whole process that has generated this tree. You don't know this whole tree. You only know the reconstructed phylogeny, which is the tree spanned by the yellow circles. Okay, and so the mathematical problem that is laid by this question is how can you compute the distribution, the law of the reconstructed phylogeny, knowing the ingredients of the model, birth and death rates, and how they depend upon the variables I just stated. It's a very popular approach, as I said. Here you see 4 survey papers that have been written about this subject. There have been numerous methods that try to infer the dependence of speciation and extinction rates upon a binary trait for that's BC, a quantitative trait, quasi, GOC for geographic trait, hidden trait, etc. And we have the group has contributed to this topic through this list of papers, and mainly our let's say our specialty was dependence upon time and a non-heritable trait like age. But of course, the question that you may ask is how much signal is there in a single phylogeny, right? To understand the diversification process, you just assume in this approach that you're given one single tree, okay? And so my take on this topic now is that we should add more signal. Some use fossils, and I will claim that we should use the additional signal coming from genomes. Okay, actually, species trees are most of the time in recent years have been inferred thanks to molecular data, and so phylogenies are called molecular phylogenies. And actually, what happens is that the sequences that we use to build the trees at each gene. So we have gene trees at different genes. That some gene trees have the same topology. So here you have A, B, C are 3 species. You have a green lineage and orange lineages correspond to 2 different genes, and so you see that these 2 genes have the same topology as the species tree, but sometimes you have gene trees that do not display the same topology as the species tree, and here you see that B and C, sorry, A and B are closer to each other. If you look at the gene tree, the orange lineages, they're closer to each other, whereas in the species tree, it's B and C that are closer to each other. Okay, and so the deep coalescences, as you see on this picture, are due to the intrinsic randomness of coalescent, or it can also be due to spatial structure. So assume that you're given the species tree, and now you follow back in time the orange lineages, the gene lineages, and here what happened is that the 2 lineages ancestral to B and C did not coalesce inside the edge, which is the mother to B and C, and so then they find themselves with the lineage coming from A together in the same ancestral species to the 3 species A, B, C, and then what happens is that the 3 topologies are equally likely. And so this is called incomplete lineage sorting. That's for deep coalescences, and shallow coalescences are due to gene flow between species, as you see on the cartoon, where here the ancestral species to A and B, after their differentiation, have been able to exchange gene, that is to hybridize. And actually, it's a phenomenon that is quite well known now, and that was on the cover of Science in 2016. Maybe not the cover, sorry, but a paper, an article, a review article, exactly on this question of hybridization between different species, and how it messes the picture that we have of the phylogeny. So of course, so yeah, maybe I should be a little more. I should insist a little more. So what happens is that we have many genes. We can infer their topology by comparing the sequences at each gene. We infer gene trees, and of course, if all these gene trees had the same topology, this would be the species tree. Okay, but most of the time, as I tried to show in the previous slide, these gene trees have different shapes, and so there is a whole industry which is called phylogenetic reconstruction, which consists in reconciling all these gene trees. And the framework, the paradigm that is used to couple these gene trees is called the multi-species coalescent. So the idea is as follows. So you start from the species tree. So again, here, A, B, and C, for example. Of course, most of the time, it's more like trees with 1,000 or 10,000 leaves. Here, just 3. And so you start from the species tree, maybe fixed or random, like a birth death model, like in the phylogenetic approach of diversification. Sometimes, and most of the time, it's actually the unknown that you want to infer from the gene trees. And the important assumption is that conditional on the species tree, gene trees follow independent coalescence, constrained to reside within the species tree. What does it mean? It means that you consider all the lineages for each, independently for each gene. So here, a different color. So for example, light blue, you follow the lineages of the light blue lineages. And once the lineages are in the same ancestral species, they coalesce at constant rate, okay? So that's basically like a censored coalescence. And sometimes these models allow for, let's say, horizontal pipes here between two species where gene lineages are allowed to travel, but usually these events are one-off events and they're rare. There is no explicit modeling of physical linkage or recombination. Gene trees are all independent, conditional on the species tree, and there's no information coming from their proximity on the sequence. And so here, disagreements among gene trees are mainly due to the randomness of the coalescence process, which is really a nuisance parameter, which is something that let's say blurs the picture or the history of the species tree. And so all the signal on diversification in this approach is contained in one summary tree. Okay, so there are two sides of the same coin. The way you build phylogenies is using gene trees that are called molecular phylogenies. And now phylogenetic approach to diversification forgets about the gene trees, just considers the species tree and infers the diversification process from the species tree. So the shortcomings of this approach, the first one is that there are false positive associations. For example, there are authors who try to detect a dependence on speciation rates to the number of letters in the name of the species, and they found some associations. There are identifiability issues, something that has been very well described in this paper by Luca and Penel 2 years ago. Second shortcoming, species are particles. Okay, how can you? And so my take is that my question is, how can you make inferences about the processes that lead to speciation if you consider that species are particles? Speciation events are instantaneous. Okay, birth events in the birth-death model. And the only data is the species tree, assuming that the gene trees shape the species tree. Okay. So my question is whether we can use additional signal from genomes, because we know that gene trees at different loci have different shapes, to infer macroevolutionary processes without summarizing them into the species tree. Okay. So we need models coupling diversification and molecular evolution, the evolution of sequences, acknowledging various ingredients or various assumptions or features of the diversification process. The first one is that species are not particles, but made of elementary constituents, conspiring to impede or promote speciation in a quite complex way. That speciation takes time, it's not instantaneous. That species divergence is shaped by genetic differentiation and not the other way around. It's not the species tree which shapes the gene trees, but it's really the evolution of sequences that is responsible for the parental relationships between species. And that gene flow doesn't come in like one-off events, but is really continuous. We know that today species that are separated by only, let's say, 1 million years or a few million years can exchange genes. And so in the past, it has occurred, of course, continuously. And gene flow and spatial structure are important causes of gene tree disagreement. These differences between gene trees are not only due to the randomness of the coalescence process. Of course, if you assume that there is gene flow between species, as on the picture on your right, where we have sampled 2 genomes and colors correspond to different loci, you see that these gene lineages in the past have traveled through different species. And so if you think of an ancestral species as a species that has harbored in the past one or few or several gene lineages ancestral to your sample, then you see that at any point in time, the number of ancestral species to one single genome is not one. Okay, you can have several species that are ancestral to one single genome, and so you lose this idea of a species tree. All right. So the first model. So my goal here is to show you like 2 or 3 other models than the multi-species coalescent that couple gene trees without a species tree, and that takes into account gene flow. Okay, so I have other works on really the 2 or 3 first points. The fact that we could model really what happens inside the species and that speciation takes time. But here I won't talk about this. I will only talk about the extension to the species, to the usual standard approach, acknowledging that gene flow is important. So the first model is the following one. So here you see that time is flowing from left to right, okay? And so you assume that you have capital N species, which are embodied by these, these gray pipes. And the number of total of species doesn't change through time. And you will assume for simplicity that each species harbors one genome. They all, with this idea that all individuals of the species harbor exactly the same genome, right? They're clonal to each other. And this genome includes small n genes, okay, indexed by 1n, corresponding to the colors here, that n equals 3 in the cartoon, corresponding to a genome with 3 loci. And you have extinction recolonization as in Moran model at pairwise rate 1. So you see, for example, here that you see that here you had a death of one population or species, and at the same time you had a recolonization of the site left vacant by this species. Okay, and so you keep this way, you keep the number of species constant, and now you will assume that at each birth event or each speciation event or each recolonization event, you replicate the genome that was in the donor species in the target species. Okay, and now you will assume that you can add introgression to this picture. So of course, usually what happens is that introgression is due to hybridization between here this species and this species. Okay, and so what happens is that if the hybrids live in this population, then you will see a chunk of the genome which will travel to this target species, target population. But what will happen most of the time is that the genetic material that was introgressed will be eroded by recombination and by genetic drift until maybe only a small number of the lineages of the genes that were in the donor species will remain in the target species. Most of the time, it's actually zero fraction of the introgressed material that will remain in the species. So here, to simplify, we assume that each time you have an introgression event, it's only one gene that moves from the species to another and replace its homologous copy in the target species. Okay, sorry about that. And so here, what you see is an example of this process. So don't pay attention that there is shading of the colors. It's for the backward picture. So don't pay attention to the shadings. And so you see that you can follow forward in time the genomes in each of these species. Okay, you have these extinction recolonization events, and you have these introgression events. You see one here, you see one here, you see one here, etc. Okay, and so what you can do now is to sample at present time one single genome and follow its ancestry through backwards in time. Okay, and if you do that, what you get, if you look at the ancestry of one sample, what you get is that you will have a partition at any point in time, a partition of one n, where a block of the partition is the set of gene lineages that are in the same species. Okay, so here, for example, you have all your genes in the same species, and now suddenly you have one introgression event. You encounter one introgression event, and when you observe this, you create a singleton, right? Because you know that this guy came from another species, and so this and because you have a large number of capital N of species, this guy will find itself alone in the donor species. Okay, so you have now two. So the red and blue are together, but the green is alone in its species. Okay, so what you get in backward time is that you have coalescence at rate 1 per pair of blocks of your partition corresponding to birth, like this here. This event is a coalescence between the block here, green alone, and blue and the red in the same block. And erosion and the introgression can be expressed in terms of erosion, which is the fact that you have new singletons arising at constant rate C. Okay, so if you look at what happens for the number of blocks, so the number of blocks here is exactly the number of ancestral species. Okay, so you have one species at present time, and because of introgression, as you move backwards in time, you will see various species which are ancestral to your genome. Okay, of course you don't want this number to be too large, and to compute this number in terms of the parameters of the model, you can do the following approximation. So the number of blocks goes from k to k-1 at rate k choose 2 because of the coalescences. Okay, so here you have number of blocks decreasing by 1. And each time you have an erosion, you have a new block, right? Because here you had one block, and here you have, because of this erosion, you have two blocks. Of course, it's not true for the singletons. Singletons, when they undergo an introgression event, they don't create a new block, they stay a singleton. So if you forget about the singletons, you have increment of the number of blocks at rate C times N, where N is the number of loci in the genome. Okay, and so if you equal the birth and the death rate, you will see that the number of ancestral species as t goes to infinity will be a square root of n. Okay, so the problem with this square root of n is that it's quite large as n grows. If you have 10,000 loci, it means that you have of the order, if C is of the order of 1, you have of the order of 100 ancestral species. And of course, this is due to the fact that here, introgression is always permitted. Okay, we always permit introgression regardless of, let's say, the differences or the differentiation or the divergence or time since recent common ancestor of the target species and the donor species. Of course, in reality, it does happen. There is no introgression between, I don't know, like for example, a horse and between horses and monkeys. Okay, so we need to devise a model that takes into account this fact. Okay, so maybe just to go a little further with this model, so the model that studies here erosion plus coalescence at pairwise rate 1 is called Kingman's coalescence with erosion, and it's this object that we have studied with my colleague Emmanuel Scherzer and our PhD student Félix Foutel-Rodier. So I would like to be a little more precise about what I said about the square root of n. So if you look at the number of blocks, you you write it like this: number of size of a cardinal of pi n of t, which is the number of ancestral species to the sample genome t time units ago. Okay. So that I just said that, okay, the number of blocks goes from k to k minus 1 at rate k choose 2, and from k to k plus 1 at rate c times n, of course, modulo the singletons. So here I just said this, but now I want you to look at the cartoon. So here you have your blocks, okay? It's not lineages, right? It's the blocks at time 0 of your, or at any time of your partition, okay? And now you want to know how many genes there are in each block, okay? So how many genes there are in each ancestral species. What you can do is that you can see that actually these the number of I'm sorry, yeah, the origin of a block can be traced back to its singletons. So each time you have a singleton here, which was here because of erosion having arrived at some unknown time. Okay, you don't pay attention to this, but you see that the singletons in this picture. So here now it's forward time for. I'm sorry, I was confused, and so I confused you. In this picture, you see that time goes from left to right, but now it's backward time. It's the time of the Keyman coalescent with erosion. Okay, so all these coalescences here are really the coalescences of Keyman coalescent. You don't see the erosion here because we only study the blocks, and we only study, let's say, the size of the blocks. And what you see here is how singletons that are present, so dust, called dust in the fragmentation theory, how dust, agglomerate to form larger and larger blocks. Okay, so here what is not indicated here is that here, if you had like C3 and C1, which were blocks here of the partition, here you have your new block of size C3 plus C1, and now C3 plus C1 plus 1, because here you have one singleton, and so you know the size of this block because of this let's say configuration of coalescences and agglomeration of singletons. And so when K particles are present, now each particles, if you look at this backward in time, so we've looked at it backward in time twice. Each part, so if you look what happens backward in time, when K particles are present, each particle gives birth here at rate Cn over K, because Cn is the rate at which you observe erosion. But so of course, I'm sorry. Yes, exactly. But 1 over K is because you have a probability 1 over K to be the block that is concerned, and that's at rate K minus 1 over 2. And the two rates at stationarity have the same order of magnitude, square root of Cn over 2. And so what you can derive from this, conclude from this, is that the size, I mean the structure of this tree here is a critical birth and death process. Okay, and so the conclusion is here. If you set Mn of K the number of blocks of size K in pi n of t, okay, so you look at all the blocks that have size K, and now you look at Mn, the total number of blocks at stationarity. Then as n goes to infinity, you see, as we had with our informal argument, your heuristic argument, that Mn over square root of n converges, actually converges almost surely, because this I'm sorry, this partition is exchangeable and is sampling consistent. So we can define all these processes, we can couple them in n, and so it converges to square root of 2c. And now what you conclude from this is that Mn of K divided by the number of blocks goes to the probability that J equals K, where J is the total progeny of a critical birth-death process, okay? Because the size of this block here is exactly the number of leaves of this tree, which is a critical branching tree, okay? And so this is, of course, a probability distribution that is well known for a critical birth-death process. I don't give the details here. And so also what you can, what we did also in this paper, that we prove that pi infinity of T has no dust. So despite the fact that there is constant production of dust because of erosion, these small particles are immediately agglomerated to the other blocks or together, so that actually there is no dust at any fixed time. Okay, and as T tends to infinity, and when n is large, you see that you have a convergence to infinity of the number of blocks, and we also characterize explicitly the block frequencies of this partition. Okay, so I'm sorry if this was a little confusing. Just to recapitulate, we have a process where blocks merge by pair of two. Sorry, merge pairwise at constant rate, and singletons that escape from each block at rate c, and we studied the stationary distribution of this process, which is the number of ancestral species to sample genome at stationary t, like in the distant past. So here the takeaway from this is that for such a process, the number of ancestral species to a genome with n loci is of the order of square root of n, and this is not realistic. So what we did is to propose another model which takes into account the fact that in progression, gene flow between different species decreases with their divergence. So we need four minimal ingredients to generate such a model. First, we need that genomes accumulate mutations, giving rise to new alleles. Okay, so we need genetic differentiation to shape the gene trees and to then drive the ability to exchange genes. Of course, we need populations to replicate. So like in extinction recognition model, like in the previous model. Each population may be subject to genetic drift if we allow for polymorphism inside each species, but we don't have to. And now populations will exchange genes with a probability which is, let's say, proportional to their similarity. And so here what we did is to say that the probability of exchanging genes is equal to the degree of co-adaptation of the introgress gene with the target genome. So what you see here, so you have these colonization, you may have genetic drift inside the species, you have genetic differentiation by mutations, and here you see gene flow. Gene flow means that this gene will want to travel inside this other species, but will be accepted only if it's well co-adapted with the target genome here. And what is co-adaptation? So we say that the new allele is co-adapted with all the alleles present at other loci in the same genome where it arose, but is not co-adapted with all other alleles that it has never seen. Okay, and so by doing this, what we do is to propose a simple model coupling gene trees, neutral with no explicit modeling of physical linkage, but without a species tree, and explicitly modeling how the progressive loss of co-adaptation by genetic differentiation, let's say, provokes reproductive isolation in the long run, and so the fact that species differentiate definitively, and exchange genes and no longer exchange genes. Okay. Of course, this such a model is prohibitively slow for simulation or inference, and so we wanted to devise a similar model, but in the coalescent fashion. So here, what we did, so we need to derive a new notion of co-adaptation in backward time. So you remember that in forward time, gene lineages accumulate mutations, giving rise to new alleles, that a new allele that arises by mutation is co-adapted with all the alleles present at other loci in the genome where it arose, but is not co-adapted with other alleles that it has never seen. And so what we see here is that co-adaptation in forward time, you see that new alleles are co-adapted with genomic background, that's by assumption, and that separately evolving populations progressively lose co-adaptation of their genomes by mutation and their ability to exchange genes. Okay. Now, in backward time, what we did is to say, okay, a gene lineage is ancestral, can be ancestral, as the blue one here, to several genomes sampled at present time. Okay. And what we assumed is that, or how we define co-adaptation is that two non-homologous gene lineages. So if you look, for example, at the red and the blue here, they are non-homologous because they don't carry the same color. They are co-adapted if they are both ancestral to one common genome. So here, the blue lineage here is co-adapted with all the lineages here and all the lineages here. Okay. So in backward time, co-adaptation increases as coalescences occur. And the idea is that because these lineages, because this blue lineage has descending lineages that find themselves in the same genome as these guys, it means that they can't have they cannot have accumulated too many mutations, and so to be able to be harbored by the same genome. And so because of this, what we will do in the model is to impose that co-adapted gene lineages will have to coalesce more rapidly than if they had never seen each other. And so that's here how we do that. We assume that what is called non-homologous attraction between co-adapted gene lineages. Okay, so if you look at gene lineages that are ancestral to one common genome, you will assume that they can see each other and that they are attracted to each other and go back and go into the same species. Okay, so here you see one example of a lineage attracted by another one. For example, I think the blue one. So here, the greenish one is attracted by the blue one because the blue is ancestral to has descendants in all genomes, and this ensures that your gene lineages that are ancestral to the same genomes do not spend too much time in different species. Okay, and so when A is large, most gene lineages will be concentrated in a few species. Of course, you may have a lot of others in other species, but you will have some, like a few ancestral species containing most of the genome. Now, trait B, you assume homologous attraction, which means that these like the lineages of the same color are attracted towards each other, and this tunes the speed of reproductive isolation. When B is large, you will have a short wait between speciations, but I put speciations between commas because there's no speciation properly defined here. You can also assume that when two lineages of the same color are in the same species, they coalesce only after a random time, exponential time. And so when C is large, there's no incomplete lineage sorting. And at trait D, you will have erosion, like in the previous model, meaning that some gene lineages escape the species that carries them, and this rate now tunes the number of populations able to exchange genes. When D is large, you will have more erosions, and you will see more ancestral species. Okay, so you have you have again 4 parameters, but you have a very easily simulable model, and which I won't show this here, but which reproduces the patterns of the forward in time model. So a few comments about the advantages of this model. First is that it's coalescence based, so it's fast to simulate. It's plastic, meaning that you can change the model by incorporating time inhomogeneity. You can assume that there are gene-specific rates. You can assume that there are genes of speciation, like the genes that are attracted to each other. There are many things that you can do. And here now the number of ancestral species to n genes grows very slowly, much more slowly than in the previous model. It grows like log n. Okay, so we were satisfied with this model because regardless of the values of the parameters, the increase with the number of genes of the number of ancestral species is very slow. It is sampling consistent, meaning that if you look at the ancestry of a genome of n+1 genes, and now you remove the n+1 gene, and you look at the ancestry of what remains, it has a certain distribution, and this distribution is exactly the same as if you had started with n genes. Okay, that's called sampling consistency, and that's something which is quite important when you model biological phenomena. So here you see, sorry, in these two simulations here, you see when n is large, you see that the numbers of ancestral species grows quite slowly, and you see that the number of ancestral species grows linearly with d over a. You also see that here the number of ancestral species, when you take, say, k equals 20 genes, and you just subsample, you always see the same thing, regardless of the total size of the genome. So I have just like one paragraph that I can go back to if you want. It's just to say that the process that describes the ancestry of one single genome in this model the right click. Sorry?
We lost you for 10 seconds or so.
Okay.
At least I.
Okay, so it's just I was just saying that it's a small paragraph that I might go back to if you want, explaining that the ancestry of one single genome summarized to the partition of gene lineages into different species is exactly the same as the Moran model with mutations under the infinite allele model. And so if you look, so you have one sampling formula that gives you this partition of gene lineages into different species. And so you can use this, all what is known about UN sampling formula and the Chinese restaurant process to derive some of the properties here. So we tested this model with two phylogenies, small phylogeny of 6 bear species and 6 finch species, only a small number of loci. These phylogenies are known to include species that have exchanged genes in the recent past. And so what we did is that we devised a kind of ABC inference method where the statistics of interest, the summary statistic, is the mosaic of gene trees that we get, and actually the pairwise distances between these gene trees. Okay, what we did is for each gene tree, we compute for each pair of gene trees, we computed the distance using either the Billera-Holmes-Wortmann distance or the Kendall-Colline distance. And then we compare the distribution of pairwise distances to the simulations, and then we optimize this divergence Kullback-Leibler divergence over the parameters. That's here. And so we recall that so A is non-homologous attraction. It tunes the time scale of gene flow. B tunes the time scale of reproductive isolation. Let's say the length between the time between nodes of the phylogeny, if there was a phylogeny, and D tunes the number of ancestral species. Okay, and so what we did is to look at. So here are the inferences that we get, inferred values of B here, of D over A here, using the BHV distance between trees, and here the KC distance between trees, and what we got is a better result for the KC distance. So that's what we use here with the data. So what you see here, so forget about this for the moment. On the right, what you see is the Kullback-Leibler divergence for when you compare simulations with this value of B and this value of D over A and the data set. And you see that the likelihood surface is very well behaved with here maxima here and here. That's for the two different, here the Bayer data set and here the Finch data set. Whereas when you look at the MSC model, the multi-species coalescence, you see that the likelihood surface using the birth and death rates is not well behaved, and so it's very hard to find a maximum to let's say to convince you or to show you why this is true, that this model does much better than the multi-species coalescence. It's because of this. So here what you see is the distribution. So each breakdown here is the distribution of pairwise distances between gene trees. Here for the Bayer dataset, here for the Finch dataset. The gray part is the same here and here, the same here and here, because it's the data. Okay. And in blue, what you see is the simulations that were done with the best fit parameters. Okay. And so you see that the distribution of pairwise distances, the distribution of pairwise distances is much closer to the real one with our best fit simulations under our model than under the MSC model. And why is this true? It's because the MSC model can only explain the discrepancy between gene trees thanks to incomplete lineage sorting. And so you see that under the best fit parameter simulations, you see that you have to have like a birth and death speciation extinction rates that are such compared to the time scale of coalescences that you have many genes that are very different from each other due to many very deep coalescences. Okay, and so what happens is that the deep coalescences are not sufficient to explain the discrepancy between gene trees. And here you see that in this model where we completely forgot about deep coalescences and only model gene flow, we do much better. So of course, now we could even improve this fit by allowing for incomplete lineage sorting, but here it's not even included. So my conclusion is as follows. So here's the comment linked to these figures. Overestimation of the role of incomplete lineage sorting in the discrepancy between gene trees produces a very bad fit. Of course, there's a mathematical question. Does the gene-based diversification model that we devised in backward time have a nice dual in forward time? You have some perspectives like to take advantage of whole genome sequences instead of just a few loci. And instead of inferring the parameters of the model, infer the whole entangled macroevolutionary history of these genes. And here my conclusion is that models coupling diversification and molecular evolution are only in their infancy, that there's a lot of room for people who want to model diversification and molecular evolution to try to join efforts to do a better job than just the multi-species coalescence. So here a small acknowledgement of, sorry, not a small, very thankful acknowledgement of my co-authors, and in particular Félix Foutel-Rodier and Julie Marin, who did all the work. Félix is a postdoc now in Montréal, soon at Oxford, and Julie is a maître de conférence in Paris Nord. And just a few slides, just two slides. The first one about peer community initiative. We have founded a couple of years ago the peer community in mathematical and computational biology. The papers that are recommended, meaning reviewed by independent reviewers, can then be published in the journal of your choice, but can be also, if you want, published directly in the peer community journal. And this is free for authors, this is free for readers, free for everyone. So please support us by submitting your best papers to this platform. And of course, don't forget that we are in a climate emergency. Thank you very much for your attention.